Hello everyone, I welcome you all. Today we are going to learn about fermentation process of sugar using yeast. For this experiment we will need all these things listed here, and please do not forget the adult supervision as well. So let's start the experiment. First we will be going to add 1 teaspoon of yeast in each bottle using funnel, so that it do not spill outside the bottle. To do this experiment, I have made funnel using paper. You can also do the same in case you do not have any funnel with you. Also, the yeast which I am using here is dry yeast, which one can easily get from the grocery shop. Next, we will add sugar. Now watch carefully. We will be going to add no sugar in bottle number 1 teaspoon of sugar in bottle number 2 and 3 each, 2 tablespoons of sugar in bottle number 4. Next, we will add 1 glass of lukewarm water in bottle number 1, in bottle number 3 and in bottle number 4. In bottle number 2, we will add 1 glass of boiling water. Add boiling water carefully. You can do this with the help of an adult. Instead of adding boiling water, we could have also boiled the content of bottle number 2. Now close each bottle with the help of its cap and shake it well so that it gets mixed properly. You have to shake the bottle carefully so that you do not spill the water outside the bottle. Remove the cap of each bottle and close the mouth of each bottle with the help of a balloon and leave it for few hours. During this time, observe any changes which are taking place and note it down carefully. So, are you noticing any changes? Definitely, there are some changes, right? Before explaining you the reasons behind those changes, I will going to explain you about fermentation process. Then you yourself will be able to understand and explain the reasons behind those changes. Let's first start understanding what is fermentation. Fermentation is a biological process in which carbohydrates such as sugar, starch, etc. are converted into either alcohol and carbon dioxide or organic acids with the help of microorganisms like yeast, bacteria, etc. Now the question arises how we came to know about fermentation process. So, fermentation is a process that has been known to humanity since thousands of years, mostly in the form of fermented beverages made from fruit, rice, etc. But people were not aware of the basic science behind the process. It wasn't until the mid 1800s when a French microbiologist, Louis Pasteur, who connected yeast to the process of fermentation. He originally defined fermentation as respiration without air. There are many types of fermentation process named depending upon the product formed in the process. Here we are learning about alcoholic fermentation. Let us understand what happens in alcoholic fermentation. Carbohydrates act as a food for microorganisms such as yeast. Yeast depend on carbohydrates for their energy. In this process, yeast break down sugar in order to be used as energy. And while metabolizing their food, that is sugar, they produce carbon dioxide and ethanol as waste products. One might ask, what it does have to do with the blowing of balloon? It definitely does, because it is this released carbon dioxide gas which causes the blowing of balloon. As soon as carbon dioxide gas is released, it increases the pressure inside the bottle and eventually comes out of bottle and causes the balloons to expand. If this is the case, then why aren't balloons on bottle number 1 and 2 are blown? It's because there was no sugar added in bottle number 1 and hence no food for yeast to metabolize and release carbon dioxide gas. Then what about bottle number 2? We added sugar in it, right? Yes, but if you remember, we also added boiling water in it, and fermentation process can happen only at certain temperature, also known as optimum temperature. The optimum temperature for fermentation process is usually around 32 degrees Celsius to 35 degrees Celsius. Since temperature of boiling water was more than optimum temperature, hence enzymes were deactivated, that is got killed and could not initiate fermentation process. Then you might ask why the size of balloon on bottle number 4 is bigger than that on bottle number 3? Good question, I must say. It's because we had added 2 teaspoons of sugar in bottle number 4 whereas only 1 teaspoon of sugar in bottle number 3. Due to more food available for yeast in bottle number 4, it releases more carbon dioxide gas, which in turn causes balloon on bottle number 4 to be blown bigger than that on bottle number 3. Let us proceed to the applications of fermentation. Do we have any applications of fermentation in real life? Yes, we do. There are many, right from making those fluffy bread, production of wine, to making those tasty yogurt and fermented pickles. All these processes involve fermentation. I hope you understood the science behind the fermentation process. Thank you so much for watching. Hello everyone, I welcome you all. Today we are going to do experiment through which we are going to learn about convection mode of heat transfer. So let's get started. To do this experiment, we are going to need 2 glasses, some cold water, some hot water, spoon, any color, a CD, any card, and yes, most important, adult supervision. To start with the experiment, take 1 glass and pour some cold water in it. Now take another glass and pour some hot water to it. Do it carefully so that you do not spill on your hand. After this, add some color to the glass containing hot water. You can add any color as you like. After this, with the help of a CD and a card, cover the glass containing cold water and very carefully place it on the glass containing hot water. After placing the glass, remove the card very carefully in order for the transfer of heat to take place. You can see here that as soon as I remove the card, the hole in the CD allows the hot water to rise up, which can be easily seen by the movement of colored water in upward direction. Now let's do the opposite and see what happens. To do this, take another glass and pour some cold water to it. Next, take another glass and pour some hot water to it. Now add some color to the glass containing hot water and mix it well. Place the card and a CD as a lid on the glass containing hot water and very carefully invert the glass containing hot water and place it on a cold water containing glass. Very carefully you have to do it. After placing the glass, remove the card very carefully so that you do not spill the water. As soon as I remove the card, do you see any difference as compared to earlier? Definitely yes. As you can see that this time the colors do not mix. That is, hot water is not moving downward. What may be the reason for this? The answer lies in convection mode of heat transfer. So let us now study what exactly is convection mode of heat transfer. As we all know, there are 3 modes of heat transfer: conduction, convection and radiation. Today we will focus on convection mode of heat transfer. Convection is defined as the process of heat transfer which occur in fluids from an area of warmer region to cooler region due to temperature difference occurring between two parts of fluids. Convection occurs only in liquids and gases because molecules within liquids and gases are free to move. You might ask, if convection is transfer of heat from warmer area to cooler area, then why in second case transfer of colored hot water from upward to downward direction did not take place? The answer of this question lies in convection current. Let us now see what exactly convection current is. Here we are going to understand what is convection current and what exactly happening in our experiment. Due to temperature difference between different parts of the fluid, hot fluids tend to rise whereas cold fluid tend to sink, which create a current within a fluid known as convection current. When a fluid is heated, it expands and becomes less dense. This less dense heated fluid rises away from the heat source and pulls down cool fluid in order to replace it. This cooler fluid in turn gets heated and rises and again pull down more cool fluid. This cycle continues and stops only when heat is evenly distributed throughout the fluid. This is exactly what happened when cold water glass was kept on hot water glass in first case. For second case, as we know, hot water is less dense than cold water, hence it does not sink in cold water, that is moved downward. This is what happened when we inverted the position of glass in second case. Therefore, we can conclude that convection mode of heat transfer take place only in upward direction and not the other way round. Let us see where do we find the use of convection in our daily life. There are many uses of which I have listed down a few. Have you ever given any thought why generally heaters are kept closer to the floor and air conditioned higher up? Here convection come into play. We can heat or cool our house more efficiently if we keep convection in mind. Since hot air tends to rise and cool air tends to sink, it makes sense and helps to put heater closer to the floor and air condition higher up. Same is the reason why freezer unit is placed on top of refrigerator and not at the bottom. Similarly, hot air balloon is also an example of convection because a heater inside a hot air balloon heats the air, causing the air to move upward. This causes the balloon to rise. So that's all from my side about explanation of convection. I hope I made it easy for you to understand the science behind convection mode of heat transfer. Thank you for watching. Hello everyone, I welcome you all. Today we are going to learn about chromatography and believe me, it will going to be colorful. To do this experiment, we are going to need few sketch pens, few white chalk pieces, a plate and water. So let's start the experiment. First we will draw around each chalk pieces using sketch pens as shown here. I have already drawn it. After this, we will place these chalk pieces on a plate in a standing position. After doing this, take some water and pour it on the plate carefully. You can see as soon as I pour water, the color of the sketch pens has started separating out on chalk pieces. If we observe after some time, then we can see that all the colors of a particular sketch pens have got separated out. Let the chalk pieces dry completely and observe it carefully. We can see that even though the ink of sketch pens appear to be made of single color, but it is actually a mixture of many colors hidden in it, which can be seen in the form of separated colors of a particular ink. The separation of colors of a particular sketch pens have been possible due to a process known as chromatography. Let us now understand what is chromatography and basic principle behind it. Chromatography is a technique used for the separation, identification, and purification of the individual components of a mixture for qualitative and quantitative analysis. The technique of chromatography had its origin in the pioneering work of F.Y.B.Ed, who separated plant pigments such as chlorophyll using packed glass columns. When we talk of principle of chromatography, we can say that it basically exploits the relative affinity of the individual components of the mixture to the stationary and mobile phase. It is based on the principle where individual components in mixture, when applied onto the surface of a stationary phase, is separated from each other while moving with the help of a mobile phase. Let us now understand what is meant by stationary phase and a mobile phase. Here, stationary phase is always solid phase or a layer of liquid absorbed on the surface of a solid, and mobile phase is always liquid or a gaseous phase. If you are finding principle of chromatography difficult to grasp, then let me make it easier for you. Think of chromatography as a race. Waiting on the starting line, we have got a mixture of chemicals in liquid or gaseous form. When a race starts, runners soon spread out because they got a different abilities. In the same way, individual components of mixture spread out because they travel at different speed over a stationary phase. If we go into the advantages and applications of chromatography, then there are many, of which I have mentioned here only a few. Chromatography requires very low quantity of sample for analysis and can work on a wide range of sample. It can also separate components which have very similar physical and chemical properties. Due to these reasons, chromatography is considered as a novel method and a very powerful tool in the hands of chemists. Chromatography finds application in many diverse fields such as analysis of foods and drugs and also in forensic investigations. I hope you understood the basic science behind chromatography. Thank you so much for watching.